In this video, you will learn about protein synthesis, how proteins are made. But first, I will explain to you the components of DNA, short for deoxyribonucleic acid, messenger RNA, and transfer RNA. These are called nucleic acids. A DNA molecule is made up of two rows of nucleotides that form a ladder shape. This ladder is twisted and is called a double helix. Each nucleotide in the DNA molecule has three parts: a 5-carbon sugar, which in DNA is deoxyribose, a phosphate, and a nitrogen base. There are 4 types of bases: adenine, which is referred to as A, thymine, which is referred to as T, guanine, which is referred to as G, and cytosine, which is referred to as C. Adenine will only bond with thymine or uracil. We'll get to that later. And guanine will only bond with cytosine. As a result, adenine is always next to thymine, and guanine is always next to cytosine on the DNA ladder. There are exceptions to this, but those exceptions will not be discussed in this video. So DNA is a nucleic acid made up of deoxyribose, phosphates, and bases, and its name is short for deoxyribonucleic acid, which makes sense because it's a nucleic acid and has deoxyribose. Messenger RNA, also referred to as mRNA, is the same as DNA except that it is a single strand of nucleotides, has uracil in replace of thymine, and has ribose in replace of deoxyribose. Also, each group of 3 adjacent nucleotides is referred to as a codon. Transfer RNA is the same as messenger RNA, except that it is always 3 nucleotides long. It is often referred to as tRNA for short. Now we can discuss protein synthesis. There are 2 steps in this process: transcription and translation. During transcription, RNA polymerase unzips a piece of DNA and attaches messenger RNA nucleotides to one strand. This forms a messenger RNA molecule. The messenger RNA molecule then detaches from the DNA strand, then leaves, and the 2 DNA strands rejoin. Then the mRNA molecule exits the nucleus through nuclear pores and ventures into the cytoplasm. From here, the mRNA will either find a ribosome in the endoplasmic reticulum or find a free-floating ribosome in the cytoplasm. Once the mRNA molecule finds a ribosome, translation occurs. The mRNA strand travels in between 2 ribosomal subunits that make up a ribosome. A lot of stuff happens while it does this, so there is some terminology you will need to know to follow along. This is the A site, this is the P site, and this is the E site. Now we can proceed. A transfer RNA molecule with an amino acid attached attaches to the codon in the A site. Then the mRNA molecule travels a distance of 1 codon. Next, a transfer RNA molecule with an amino acid attached attaches to the codon in the A site, and the transfer RNA molecule in the P site gives its amino acid to the transfer RNA molecule in the A site. Then the mRNA molecule travels a distance of 1 codon. The transfer RNA molecule in the E site detaches from the mRNA strand, and a transfer RNA molecule with an amino acid attached attaches to the codon in the A site. The mRNA strand travels a distance of 1 codon, and while this occurs, the amino acids attached to the tRNA molecule in the P site attach to the tRNA molecule previously in the A site. The tRNA molecule now in the E site detaches from the mRNA strand. This process continues until the entire strand of mRNA has passed through the ribosome. The end result of this is a chain of amino acids held together by peptide bonds. The chain of amino acids is a polypeptide chain, which is a protein. The protein can now be used to form structures like ion channels, or it can act as an enzyme to speed up reactions in the cell. Now you might have noticed that each transfer RNA molecule had its own specific kind of amino acid attached. For instance, this transfer RNA molecule with the bases C, A, and U has valine attached to it, which is a specific kind of amino acid. This transfer RNA molecule with the bases A, U, and G has tyrosine attached to it, which is another kind of amino acid. A transfer RNA molecule with the bases C, A, and U will always have valine attached to it, and a transfer RNA molecule with the bases A, U, and G will always have tyrosine attached to it. Also, each transfer RNA molecule will only attach to a specific codon of mRNA. For instance, this transfer RNA molecule with the bases A, U, and G will only attach to a codon of mRNA with the bases U, A, and C from left to right. So in effect, each codon of mRNA codes for a specific kind of amino acid. Hopefully, you now understand protein synthesis, and if you don't, you can always watch this video again.